everybody for showing up to our Friday seminar series for conservation sciences. And thank you, Dr. Erickson, for joining us. So just a little bit about Dr. Erickson. He's a research quantitative ecologist with USGS's Upper Midwest Environmental Sciences Center in La Crosse, Wisconsin. His research focuses on applying quantitative ecology to invasive species. Currently, he is developing population models to inform invasive carp management and developing methods for applying occupancy models to environmental DNA, or eDNA. He also leads the Department of the Interior Invasive Carp Modeling Team. Erickson earned a PhD from Texas Tech, majoring in environmental toxicology with a minor in mathematics, and his undergraduate degree is from the University of Wisconsin-Stevens Point. And so with that, I will turn it over to you, Dr. Erickson.
Thanks for the chance to speak. It's great to be back. I've given a couple of seminars, I think 2015 and 2017. So it's nice, and hopefully I can come up and give one in person again as well someday. So my talk has sort of two goals. One is to talk about my research, but it's also to let especially the grad students and postdocs and any undergrads see what is a career like with the USGS, or at least my career. So I'm a research quantitative ecologist, as you mentioned in my introduction, but what does that mean? That means I use math, statistics, and computer science to help resource management. For example, I actually work a fair amount with alum from your program. Grace Lapno, who's with the Minnesota DNR now, her and I talk on a regular basis, and we develop population models to help her. I also talk with Jennifer Zemansky, who graduated maybe 15 or 20 years ago from your program, and she's an endangered species biologist, so I help her with math. For example, Jennifer and I had a call yesterday where we were talking about probabilities and conditional probabilities and how it's important for endangered species management, and what happens if probabilities are not independent and how it impacts management. So using math to help natural resource people. My story might be similar to some of yours, I don't know. I grew up in a small town, Tomah. You've probably driven by it because it's where 90-94 split. Growing up, I like to be outdoors. And you can see me at the Nacita Wildlife Refuge with some pictures from when I was a Cub Scout and Boy Scout. I'm banding ducks in one picture. I'm working with Rich King, who was then the biologist, with some snakes, and he was showing us data as well. This shows both I liked being outdoors when I was little, and I was lucky to live in a spot where I could go outdoors, but also how paths are interconnected. Rich King moved up with Fish and Wildlife Service and actually ended up with an office next to mine in my center, because he works for the regional office now. So somebody who I was peeking over his shoulder and holding snakes with now has an office just down the hall from me. So you'll never know when your paths will cross again. So I grew up in Tomah, graduated in 2002, then went to Stevens Point and Wildlife because I liked being outdoors and it just seemed like a fun thing to do. I also knew these people like Rich King who loved their jobs. Along the way, I picked up a minor in chemistry because I had a friend who suggested and said it would help with employability because the wildlife job market's tough. She went to Texas Tech for grad school and got her master's in environmental toxicology. And she graduated, but I checked out the program and got an offer, so I went there. My master's project was modeling dengue and mosquito dynamics, as well as climate change, because my advisor had a grant from the Department of Defense. I liked that project and my advisor, but I didn't want to be stuck into that project. So I decided to stay at Texas Tech for my PhD. I also picked up a math co-advisor who she told me if you want to do modeling, you have to learn math. So I took a couple of years of math leveling courses when I was there, but then I was able to do some lab study and modeling study. So I looked at how toxicants impacted Daphnia in the lab. So it was basically getting Daphnia going and then throwing in pesticides. So it was very much trying to be applied work to work with EPA and their sorts of needs for data for pesticide regulation and environmental impacts. When I was at home at my parents' house one winter, I remember the Wildlife Society and part of the biometrics working group. I saw an email come across that said a postdoc position in La Crosse, Wisconsin, working with the USGS to look at the impacts of wind energy and white nose syndrome on the Indiana bat and little brown bat. So I emailed Wayne Thogmartin back and said, here's my cover letter, here's my CV. Oh, by the way, I'll be in Tomah until like mid-January. So Wayne's like, oh, that looks good. So he actually drove over one day, picked me up, and then we met in Wisconsin Dells with somebody from Madison to talk about the project more for an informal interview. So and then I ended up accepting that postdoc as well. Interesting for my career path, there were several positions I applied for and never heard back, or only got an interview for one other postdoc. But then the one position I wanted, I got. So it's sort of serendipitous. I also had several offers I didn't want that were in Texas at Lubbock because I wanted to move away from there. If you've ever been to Lubbock, you'd know why. But just the career paths take weird things. And it was also neat because I was using the same modeling tools for my PhD as my master's project. So if you're currently a postdoc or a grad student, just think about how your tools work with other systems as well. So maybe not just Daphnia in the lab, but also wind energy and bats. Currently, my biggest project is population modeling for the invasive carp in the Mississippi River. I work very closely with state agencies and other federal agencies, and I'll talk more about this today. I also help a lot with environmental DNA studies. So Fish and Wildlife uses eDNA to help guide the management and placement of their control efforts and just know what's going on. So I help them figure out how many samples to use and where to place the samples. This is done for the upper Mississippi River as well as the Great Lakes. For example, if you saw the detection of invasive carp DNA in the Milwaukee River, they were using my sample protocol. They also use the similar ideas to inform their grass carp monitoring in Lake Erie. So one thing I really like about my job is just seeing how state agencies use the results. In general, because of my skills, I also just help people with advanced computing. So if they need help, like how do I run something a lot or more efficiently, I can step in and help, sort of an amateur computer scientist or self-trained. I also just do general statistics support for my branch because I have good modeling skills and stats skills. I just help other people. This is one of the reasons they actually hired me as a permanent position because I could help with statistics. So it's like, oh, not only can you model like carp, but you can also help other people with stats. It's also a great way for me to get publications because I am just like on there as a statistician. So if you're not familiar with USGS, there's 5 mission areas. Core Sciences does our mapping. I'm with ecosystems. Core Sciences also has a bunch of advanced computing things, but ecosystems is where I am. This is also the cooperative research unit. So Lynn is part of this, for example. We have energy and minerals. So this is some of the things you might think of, like where do I look for oil or gas, rare minerals for my maybe electric car or iPad. But it's also how do I sequester carbon as other projects. So lots of neat things there. Natural hazards is looking at things like earthquakes or volcanoes. So for example, we had one of our science centers got destroyed in Hawaii when the volcano destroyed it a few years ago. Likewise, if you live in California or the West Coast, Alaska, you can have an app where it'll tell you if there's an earthquake. Then it also monitors the vibrations to help detect earthquakes. It's something that we do with Google. And lastly, water people look at like groundwater and surface water to help monitor that and inform management. Lastly, if you're not familiar, you might be with USGS, you might be thinking, why are there all these ecology people in the Geological Survey? In the 1990s, they pulled all the scientists out of agencies like Fish and Wildlife Service and NPS, and then put them into the GS. So it's a historic artifact. It's interesting policy, but it's something that perhaps your professors could have a better discussion with who aren't federal employees. So this is just a map of the different ecosystem mission areas. You can see we're across the country. You can see La Crosse just down the river from you. And then you can also see the flag, which is Lynn and other people at the co-op. So we're all over the place. My specific center, UMSC, we're down the river in La Crosse. And this is our mission statement, just to inform resource management, but it's all over the place. And I'm going to talk about the invasive carp today, which are a big problem, especially because of silver carp both invade, but they also jump up and hit people in the face. So they're both bad for the environment as well as people's safety. One thing we do with this is the environmental DNA, which I've mentioned before, but just a quick slide. So if you're not familiar with this, you basically go out, grab a water sample and see what's there. So it's seeing what's there, it takes the lab as well. And then it becomes a problem of what's in the environment. So like what DNA and why did I find it or not find it? It becomes a 3-level occupancy model. At the top level, you have psi. So is DNA at the site? Next, did you get DNA in your sample, like that Nalgene bottle or whatever he's holding? Did his protocols and sampling capture the DNA? And last, did the lab tool find it? And just as a thought experiment, think about like, how do you design this experiment? How do you do replicates? How many sites can I survey? And at a site, how do I balance my lab replicates versus my field replicates? That's where this modeling project has helped. So conceptually, here's just a figure. So on the left, is it at the site? Is it in the sample? And then did the PCR or other tool find it? This allows us to create figures like the right that tell you if you take how many samples, what's the probability of detecting? And that's just a high-level overview of eDNA. If you want to know more, we did a simulation study, and then we followed up with a refinement study. So this is another cool part of my work. So Erica worked with Chris and I, and we did a simulation study to say, this is a comparison of sampling approaches. This is how the model should work. And then Fish and Wildlife was like, well, this is really important. So we're going to actually validate your models. So they took part of a summer season, and Erica, Chris, and I all worked together. Then a bunch of other people went out and collected a bunch of samples. So to me, this is just something really cool where my research informs management and monitoring activities. So some of my other data that I work with, if you're just curious, fish telemetry. How do fish move when they get exposed to carbon dioxide? Laboratory toxicology studies. My center still does a lot of environmental health or regulatory studies. For example, if somebody wonders if a chemical is safe for EPA approval or something, my center has the ability to look at this. Metabolomics. So we both do like genomics, so like what's the genomes of species or what's the environmental DNA, but also metabolomics. So if we're developing a new pesticide, we look at a cell line, then it produces metabolites. So that's one way to look at things. And metabolomics might sound intense to people if you're not familiar, but it actually isn't. It's mainly just community ecology applied to a different framework. So again, I'm sure a lot of the grad students and postdocs, you have a diverse tool set, even if you don't realize it. I've helped with avian ecotoxicology. For example, I've looked at loon data from parts of Minnesota, or people use tree swallows as a bioindicator species. For example, we have people at my center who are looking at PFASs, which are a big problem, especially for the Twin Cities metro area. And then in general, it's just if people have questions about numbers, they oftentimes come to me for how do I model something, and I can either connect them with a resource or possibly help them myself. To me, I really like this, and it's a really fun part of my job. So the main part of my talk is population level modeling. And the problem is that the invasive carp are spreading. One focus area has been keeping them out of the Great Lakes via the Chicago sanitary canal and sewer system. So if you're unfamiliar with the Illinois River, which you probably are if you're like me until a few years ago when I started on this project, the Illinois River was connected to Lake Michigan because Chicago historically over a century ago used to dump their sewage into Lake Michigan, but that's also where they get their drinking water. So instead they diverted their sewage to go in the Illinois River. And then this created a connection. And with this connection, we're afraid of the big-headed carp spreading from the Illinois River into the Great Lakes. Also, I gave this talk once at the USGS headquarters, or a similar talk, and I was just providing this figure, and one of the guys, Lynn might know him, JT, who's her boss, said, wait, so what's going on with the Illinois rivers? They dump the sewage in the water, then they basically bottle it back up in St. Louis and ship it around back up. And I'm like, yeah, so that's maybe Todd could help you with that jab at a beverage company. But so that's just the Illinois River. And people have thought about closing it, but it's too important to close. So there's currently an electric barrier there. But part of the management is just trying to keep the pressure down on the barrier. And if you look at Great Lakes or the funding USGS and Fish and Wildlife and other people get to keep the invasive species out, it ties directly to congressional funding and priorities. So initially we got Great Lakes Restoration Initiative money, GLRI, to focus on the Illinois River because it's part of the connection to the Great Lakes. And around the time we got it, President Obama was from Illinois, Speaker Ryan was from Wisconsin, and Michigan has a very strong congressional delegation. So that was the initial round of funding. Since then, the grass carp had become a problem in Lake Erie, and there's a representative from Michigan who is very concerned about that. So we have a second pot of money that looks at grass carp. And then more recently, the invasive carp have spread up the Ohio River and Tennessee Cumberland rivers, and now we're in the state waters of Kentucky. So Senator McConnell has given us more funding to look at carp in the other subbasins of the Mississippi River. So again, this lets you see how priorities get tied to congressional interests, and that's how we get funding to do research. It's a really neat application to look at how the government works. It's more complex than Schoolhouse Rock. And with this, the goal is just to limit their spread and impact. So, but then the questions that the managers have is, how do we control the species? What action should we take? For example, would a barrier deterrent work? Maybe at a lock and dam we do something? Or what pool should we harvest in? And then where should we do these? Like, where should we harvest? Where should we place a barrier deterrent? How important is the barrier placement? And for the Illinois River, actually the lower three pools, Alton, LaGrange, and Peoria, are much different than the upper three and all have no recruitment to occur. Whereas the top pools are small and don't appear to be suitable for the carp to recruit. So it's an interesting metapopulation system as well. So to look at this, we use an integral projection model, which is very similar to a matrix model. For each pool, we have a model that's connected by just movement probabilities to the other pools. So it starts off, there's males and females that mature and recruit. And just looking at the equation on the picture on the right helps show the equation on the left. So you can see the mouse, good. So it's the population Z prime at the next time step t+1. So then this is simply the current population of size Z at time t multiplied by a kernel that changes from Z, the current size, to Z prime, the next size. And then because it's a distribution, you integrate it over, hence the integral part of the model's name. So we have a spatially explicit invasive carp population model, C-carp. It used to be spatially explicit Asian carp population model, but when we got a new presidential administration, we stopped calling them Asian carp and started calling them invasive carp. It's a similar thing to what Minnesota's been doing for probably a decade or so. So that's just the model's name. And then there's a generalized version of this as well. I've created meta IPM where it can work with other species and systems. It models the movement between pools. So it's basically connecting them and allowing movement. So just looking at pictorially, we based the population model off of a previous paper by Tahishe from 2013. And this paper emerged because people were concerned about carp getting into the Great Lakes. So they wondered, with the Illinois River, how could we control them? So they just did a single population model for the river, but included size because they wondered what size fish to harvest. And if you want to collapse the population, you have to kill all the fish, not just the big fish. And that's important because harvest tools can really only harvest the big fish. However, we also know that the spatial dynamics are really important as well, but they didn't have that data at the time, and that emerged as something else. So we've since tagged a lot of fish, and Alice and Coulter published a paper in 2018 where they looked at the movement probabilities among pools. So I know this figure is small, it's just showing they've estimated movements between pools. So we're able to take these and combine them to be the C-carp model. So that's like the example figure on the left. We run the model for a bunch of simulations just to see what's going on, and then compare different scenarios. Also, if you just notice, it's on the log scale as well because the populations vary so greatly in size. We have a lot of data to parameterize this, and we're also now expanding to other basins. Most of my work focuses on the Illinois, or most of my presentation focuses on the Illinois River because this is what's been published and what's farthest along. But we're also working with lots of people. And that's one thing I love about my job is I work with USGS and Fish and Wildlife and state people all the way from Mississippi up to Minnesota, South Dakota to West Virginia. So it's most of the middle US. I get to work with people, which is really cool. And these are just all the different locations we have carp observation data from. So it's working, for example, Minnesota DNR and Fish and Wildlife has given us points from Minnesota. It goes all the way down to Pool 27, which is the end of the upper Mississippi River where the last lock and dam is. But then we also have the Tennessee, Cumberland, and Ohio rivers. So over to West Virginia. For most of my work today, though, I'll just talk about some of our more recent published data. So this was what we had for that paper, and this is just a summary. So we have 2 species of carp, bighead and silver, and then we have the 3 different river systems, Illinois, Mississippi, and Ohio, and different pools within. And this figure just shows, we have a lot of data. It's about 42,000, I think, for this paper fish. Now it's up to close to 47,000 fish. So for ecology, it's a really big data set. And then this just shows how it's been collected different methods. Electrofishing, the silver carp especially jump a lot. Some get harvested by nets, and sometimes people don't tell us how they've captured them. So it's just a huge data set, and then it's working with lots of people. Also, just as an aside, bighead and silver carp are closely related, and in the US, they hybridize in their invasive range. However, in their native range, they do not, nor do they appear to hybridize in Europe. So it's just, from a species genetics point, it's very interesting as well, because bighead carp seem to lead the invasion, and then silver carp seem to follow. And then they also, again, hybridize. So it's just a neat system to learn a lot from. So with this data, we get the inputs. So for example, we have a maturity curve. So what's the probability of females mature given her length? We have fit the model for different pools, and then we use it to fit a Bayesian hierarchical model. So this shows, for example, each of these curves would correspond to one of the lines here. And then we have the hyperdistribution and credible interval around it as well. This helps us with a lot of different ways. So first, it helps us when we have pools without a lot of data, we can still estimate curves for. Second, it allows us to use estimates for pools without any data, rather than just uninformed or other choices. We do similar things for length weight. So total length, total weight, and we have different curves. For example, like this pool with only 3 points, because we're using a hierarchical modeling approach, we can pool information from other locations and at least estimate a curve. And then also we fit von Lampe growth curves. So these are just example curves. You notice one challenge we have is a lot of pools don't have small fish collected. And this is something else I really like about my job is I work closely with Fish and Wildlife. So we had calls earlier this spring when they're setting up their monitoring program, like what size fish do you need for the model and what would help us make management? So we were able to say, you know, rather than focusing on a lot of pools, just focusing on the smaller fish and a few pools would be much more helpful. But we know, then we just had that conversation about where to collect fish. So it's something that I think is really cool about my job is we talk to managers and they listen. So hopefully everyone in the Cons Bio program, or Cons Science rather now, wants to have your science be used. And that's one thing I love about working with USGS is we get to build that relationship of trust with the management agencies so they listen to us for advice. So we take these with the movement probabilities, which there's actually a separate team now. So Allison did her work as a postdoc at SIU, and now she's at South Dakota State. But there's now a USGS and Fish and Wildlife team that's doing telemetry and another USGS modeler who's working on the telemetry part. So I'm not going to talk about that other than just to highlight we're a big team effort. So we fit the model for the different pools and we end up with a distribution at the end. So it's a box plot. Then we have different scenarios. So the first question is, where should we harvest? So we have a reference scenario, which is the red line. And then do we harvest in the upper 3 pools or the lower 3 pools? And given uncertainties with the model, we're only comfortable looking at upper and lower, not specific pools. If we only look at this figure on the left, we see these are the different harvest levels. So we only harvest in the upper pool. Notice, even if we were to harvest all of the fish out of the upper pool, the population still doesn't collapse. That's because the lower pool is the source and the source sink dynamics. And then if we move along farther, we see basically lower harvest is much more impactful than upper harvest. And initially this was sort of a surprise to management people because they thought, oh, we just want to harvest and kill at the front of the invasion front to keep them away. However, with the proportional harvest, it's actually more impactful to harvest the recruiting population as well, rather than only the upstream. And then we also, the blue line is about a 90% decrease in the population, or I guess it isn't a 90%, because just talking to management, that's what they said, okay, if we could reduce the population at the uppermost pool. So again, this is, so the uppermost pool is Dresden Island. So let's see, Dresden. I'm sorry, I'm getting my Illinois River confused. I need to go back to the map. The uppermost pool is, yeah, Dresden Island. So Dresden Island's population. So this is the change to the Dresden Island population. So that's their important goal, is keeping the pressure down low at the invasion front. So this is the relative change. So harvesting outside at different pools rather than the pool is important. So that's one big take home. But next is, where should we do a deterrent? And let's see. Oh, that was just Chris throwing a question in. So yeah, this is where do we place it? So we just looked at two harvest levels, 0 or 25% in either the lower or upper pools. So this would be no harvest. And then the color is where we place the deterrent. And there's a lot going on with this figure. So the biggest take home is just try to place the deterrent sort of farther downriver if you can. Is small, and then also, again, the harvest as well. So also, we can only place the deterrent reasonably at these three pools because they're high head lock-in dams. So they're similar, for example, to what you have in Minnesota on the Mississippi. Conversely, the downriver ones are wicket dams. So they're not, there's easier for the fish to pass, is a quick summary of that. Lastly, with this model, we just looked at sensitivity. So there's a steepness parameter with the growth model. So we just compared different values. We use 2.1 as our base level, then we use different harvest levels. So the two steepness parameters closest to the 2.1 we use produce similar results. So this just lets us see that qualitatively, at least, not that much is going to change if we get the steepness too wrong. So we have some limitedness. So this is just one of the types of analyses we did with the model, but it also just shows that if you're taking, for example, a population ecology class, and they're talking about model sensitivity analysis or different model analysis approaches, this actually is something that managers are interested in and care about. So take homes from this. First, the source sink dynamics are actually quite important. It's something that's why we built this model, and it's not always intuitive, especially metapopulation dynamics, to resource managers or other people who don't work with it on a regular basis. We want to limit the source by targeting downriver movement. So basically keep the fish downriver. It's sort of a numbers game where if 10% make it through each stage or something, hitting the farthest downriver hurts the numbers the most. Also limit the spread, so target the downriver. And then, so that's the technical part of my talk. The second part is just more my career with USGS. So how did I get my job? Well, a large part of it was just luck. I happened to get a postdoc with my center. At the time, 2013, sequestration was going on. So there's somebody at Patuxent Wildlife Research Center who I'd worked with, or at least talked to, and knew him professionally since I started grad school. And he really wanted me as a postdoc. but he couldn't hire me because of sequestration. However, Wayne at UMass was able to hire me as a contractor through Cherokee Nation Services because they were a no-bid minority-owned federal contractor. A year into my 2-year postdoc, however, Cherokee Nations got investigated because they were charging 60% overhead on a no-bid contract. So they got investigated by Congress. So then Wayne was able to claw back the money from them and hire me as a term. And then I was just talking to people down the hall first about eDNA, and they're like, well, how do we analyze this? And I'm like, that sounds like an occupancy model. But it turns out we actually had to use a 3-level occupancy model. So I got started with that. And then I also started doing population modeling with carp later on. So a lot of it's luck. It's also being a generalist. So if I had a very specific tool set, it might limit me, but I, at least with my modeling skills, can work on a lot of different things. I've saw several other cases of this as well. For example, when I was a postdoc, another team member was hiring a postdoc in Denver, and they had me look at the 2 finalist candidates' resumes just to see what person would we need to work with or would I want to work with, and just another opinion. One person had studied bats for their PhD and like had a really strong bat resume, and like had only studied bats, and they were an expert in bats. And this was looking at wind energy on bats. The other person, however, she had studied like brown bears for her master's or something, a different species for her PhD, and had a postdoc where she'd looked at like a 3rd species. And she had so we actually chose her because of her diverse background. So with academia, sometimes you have to be very specialized. With the USGS, having the right generalist skill set helps as well. Just another example as well, we hired an aquatic invasive species eDNA expert recently because somebody got promoted. So we chose somebody who actually had never done invasive species before, but had done a lot of eDNA with amphibians, and just came in with his research plan. So if you, for example, get like a USGS permanent talk, or like a permanent job offer, a lot of it's like, what will your research program be and how can you articulate that? It's not necessarily your past. We don't care, have you published 100 papers or 5 papers on X? We'd rather see in general, like what's your skill set and how can you use them to address partner needs and change? And last, my advisor's parting words of career advice were be needed. So for me, this has been helping people with other stats data, or it's selectively saying yes at the start of my career. If somebody needs help, sure, I can help you do that. It's not overburning myself. It's something else I've learned more recently, saying no, but it's just have a skill set that people want. So if you're thinking like skill sets for USGS and things, what skills? The 3 R's, reading, writing, and arithmetic. So just, it's a lot of reading, as you probably know as students and postdocs and faculty. We have to read to stay current in our field. It's reading reports, reading lots of things. Also lots of writing, writing papers, writing proposals, just emailing and communicating with people. And it's, yeah, all the writing's important, but luckily with USGS, we don't have to do as big of proposals as like NSF ones. For example, NSF proposals are notorious to be like 20 pages long or things. We have to do funding templates, and those are like 3-page limits each year. And then we also have internal sources where they connect us with partners. So for example, they didn't, like if you start up as a faculty member, it's like, here's your job, here's a startup package, good luck getting money oftentimes. With USGS, we do mentoring. So we have a formal mentoring program, and I was actually mentored, luckily, with my program manager in Arreston. So the person who does all the invasive species funding choices was my mentor. So she could help say, this is how the process works. This is how to get new projects going. So also I go out, for example, to Arreston, and they'll send you out there, and you might give a talk to one program, but they say, while you're there, you know, go, you're like, so my supervisor and my center director will say, here's 3 other people you should talk to. And then you build a network that way. Last skill is arithmetic. So lots of statistics and math. And yeah, just, I have to keep learning with those. So just more building upon that, it's just a lot of self-learning. So In grad school, you probably transition from your first courses to later you're doing your research and maybe have some seminars, but it's a lot of learning the skills you need. And that's something I have to do on a regular basis. Communication, it's seminars like this, but also talking to people on a regular basis and clearly explaining what's going on with science. Statistics and math I use a lot. I mentioned, I think Jennifer, I was talking to Jennifer Zemansky a couple of days ago, and she called me up because she was confused about probabilities. So I had to talk her through that so she could explain to somebody else. Biology, I wish I used more biology, but I do use it a fair amount, and a lot of it's bridging biology with other things. And programming. When I started grad school, I really had never programmed before, but my advisor had me initially do all my modeling in MATLAB, but then we switched over to R because he was switching over. And now I use R, Python, Stan for Bayesian things, Git to keep track of software, and version control. So it's just lots of learning and programming. And that's something I like, is that I just keep learning with my job and things don't stay the same. So that's about my job. More generally, if you're applying to USA jobs, here's some tips. First, everything I say is a summary or a variation of this public web page. So if you search USA Jobs Help Center, what should I include on my federal resume? Read through this. It's actually really good. Here's just some key points I've saw. Match keywords. I misspelled keywords. I put keyworks. Oops. But let's say a position, you're applying for a fish, like a biologist position that says enumerate fish. So you don't say I counted fish or I surveyed fish, or I use like a normal word choice, match it and say I enumerated fish. USGS's HR literally matches words as the first round of screening. So you need to match the position description with your own experience. There's a joke in academia where like tenure committees or search committees can't read, but they can count. So they can count your publications or count students, but they can't like read your publications or know quality. USGS's HR and general, the federal HR can use a word search, like a Control F find, but they can't read. So it's like match the words in the position, because I've had HR people show me how they go through resumes and they literally take out a highlighter, whoop, whoop, whoop. Yep, same keywords. We're passing you on to the scientist. You get to see if you're qualified. But if you have a position with questions, be generous to yourself, but also realistic. So don't lie, but also let's say it's like qPCR, and maybe you're using qPCR as a grad student, and they could say, like the position asks for your experience with qPCR, and there's like A through E, you have to rank yourself from never used it in classes to the last one's expert, and teach other people. If you've used qPCR, and even if you just had an undergrad how to use it once, you could say you're an expert. So that's a way to be generous to yourself. And then on your resume, you would include like, like research project, taught students how to use qPCR, or taught undergrad how to use qPCR. So match the words that way and be generous. We want you to be humble when we work with you, but you have to be generous to get through HR. There's a lot of entry positions that have these questions if you've ever used USA Jobs. Include all of your transcripts, including 2-year degrees, summer universities, everything. One of the professors from my grad program applied for a toxicologist position at my center. And he is a professor of toxicology, and HR said he wasn't qualified because he didn't properly include all of his transcripts or describe his courses. So our HR is notorious for this. So details are important, and that's federal HR. You might think, oh, well, I went to a 2-year program, associate's degree. Why would I include that? Well, USGS HR wants you to, and our federal HR requires it. Also, I personally took a summer class through a different university. So anytime I apply for a federal job, I have to include that transcript. So even though it's only like a 2-credit course, one summer I'm from a different university that's transfers into my main university transcript, I still include it. Define courses. If a position says that you have to have like 30 credits of biology, say I have 30 credits of biology. I have Bio 101, 102, whatever, but then also say other courses like Wildlife 301, Ornithology is a biology course because we study the biology of birds. HR will try to read your transcript and guess, like bio means biology, but they wouldn't say a wildlife course is biology unless you tell them. Likewise, just if it says like 12 credits of statistics and you've taken 15 credits, list all of your courses, just so they see you're qualified. You can do this on your resume or through their question system, but just like list it so they can easily check it off. You want to make it easy. Basically, like imagine it's your friend who's an English major has to determine if you're qualified by matching keywords. That's roughly what our HR does. And then if you don't get a position, call up HR and ask why. Sometimes they can tell you useful feedback. For example, I know somebody who applied for a position at my center and didn't get it because she's Canadian, or she recently became a US citizen and all of her undergrad and grad courses were from, or her undergrad was all from Canada, but she had her PhD from a US university. And they said, well, your transcripts aren't translated to American universities. So she had to get a certified translation. Then she applied for a different position at my center and got it. So even though it was good enough for her PhD program, she still had to get things translated. They can also tell you, if you call HR, they can tell you you're not qualified, and you can also sometimes appeal. I don't know how to do this, but I've heard about it. So this is just a way to call up and ask why. It might not be something you realize to do, but it's something that's there. Last, I just want to talk about how math is useful for other opportunities as well, especially if you're a grad student and a postdoc, you're worried about something or like, what will I do? Maybe you feel trapped in a toxic situation. You actually have more opportunities than you realize. So for example, I went to New York City and got paid to develop an online course. Outside of USGS, I got ethics approval, but it was really fun for me to go there and get paid and do that. That's the top picture. The bottom picture is Eric Eager, who was my best man at my wedding and is a good friend. He was a biomath professor at UW La Crosse. He got tired of teaching and just the workload and low pay at a UW system. So he was moonlighting and now works for Chris Collinsworth. Chris Collinsworth has a company that does football analytics, and Eric is the head of his research and operations part. So there's a picture of Eric and Chris together on Chris's podcast. Eric now got a pay raise and also gets to work with football data, which he loves. He's a college football player. But there's lots of other spots you could use your skill sets as well. So maybe you're burnt out or don't want to work for a natural resource agency or can't. If you have a strong skill set, there's lots of other people that can use you as well. And you might not get your dream job as a professor, but you might be well paid and work less hours, or maybe find a different dream job. So that's the end of my talk. So I have plenty of time for questions. I think, how do I stop? Can you kick me out, Chris, or Nick?
Yeah, there may be I think it's in the upper right, maybe, to stop sharing. Nice.
There. So plenty of time for questions about my research, USA jobs.
I would like to ask a question about kind of getting a internship in grad school. I know USGS offers graduate kind of employment opportunities and internships. I guess I'd like to hear your thoughts on those and yeah, what you think about them generally.
Let's see. So there's, I guess, why would you take the internship? So like as a student, you might take it because maybe you don't have funding, which would be a good reason to do it. I've had interns that I hired from UW La Crosse who are in their unpaid master's program. So they work for me to get funding. So one part is funding. Another reason could be networking. It'd be a really good chance to network with people. The third might be experience. So if it doesn't hurt you with grad school, I would say do it. If it's going to like add a year on, think about it. So it would probably help you. It would help you to network with USGS. So if you want to work for us, it would let you see what's there and get to know people and know what we do. If I was hiring somebody personally, if they had the same skills from their PhD or master's program as they did from the internship, I don't think it wouldn't sway me one way or the other. But if they got new skills, or it would probably help. So it's a positive, but I don't know if it's worth like adding a year onto your grad program. There's a question, is it easier to work remotely post-COVID? I would say with a caveat, yes. USGS now, for example, I have, let's see, right now a postdoc and 3 interns. And the interns are contracted through UW La Crosse, the postdoc's federal. One of the interns through UW La Crosse is currently living at her parents' house in Green Bay, which usually works great. But actually this morning she locked out her computer with her password. So I think Monday she has to drive back over to La Crosse for IT support. But in general, yeah, USGS is allowing more remote. But a lot of it depends upon who you're working for and what they want, as well as the project. I do a lot of modeling works that can be easily done remote. Somebody I work with, my postdoc mentor actually now is somebody who is his postdoc from Hawaii, and he's actually moving to Oregon because his wife got a job with the Fish and Wildlife Service and going remote. So you can be remote, but it's for like modeling things, but it's a lot of the people and who you work with. But then it's also, if I was doing wet lab or field work, then I couldn't be remote because I'd have to be like on the river doing things. So it's COVID definitely helped us, but it's not always, but it's more likely. Chris, can you see, there's another question that I missed, and hi to Peter as well.
Yeah, I can read these off too, so we can have them for the recording. So Holly asks, did you learn most of your quantitative skills in university courses, or are some of your skills self-taught? Can you recommend any resources for learning new modeling techniques?
Yeah, so what helped me the greatest was the 2 years of courses. I had to take some leveling courses and some intro grad level courses. Linear algebra as an undergrad course. So once I basically got that mathematical maturity from linear algebra, and then getting it drilled into me through a couple of grad level courses and things, it was much easier to teach myself. So if you have a good foundation quantitatively, it's much easier to teach yourself. So I taught all my Bayesian things myself. And that was just for me finding books. At the time, Mark Carey had some good books on wind bugs and jags I liked. But so I guess it's just finding the books or whatever resources you need to learn. So I personally like books, but there's also now a lot of online courses and things. But in general, it's just having a deep, to me, like getting that deep math background helped. And that's actually something I noticed with like the stats students from La Crosse, because they have a graduate degree in statistics or an undergrad in stats, they take a lot of stats courses. So it's easier for them to learn to model then. And that's why I was glad in grad school I took those 2 years, basically spent more time to learn the modeling. So I guess to answer your question, both. Does that help, or do you want any more specifics?
Sweet. Yeah, she said that is helpful in the chat. And Dr. Sorensen threw a chat in asking about one of the models, but he says, please take me last as we can talk offline.
Sure. And I'll just say that it's the thing I like about the work is I get to work with like people like Peter I've talked to, and there's lots of academics as well and researchers. So at USGS, we get to work with a lot of people, and it's fun.
Sweet. I don't see any other questions in the chat. So Dr. Sorensen, if you want to throw your question in the chat or unmute yourself, that's awesome. I guess just for my own knowledge, so I know like if you're recently started non-tenured professor, you have you're like on this tenure track and looking to get tenure. Is there kind of any equivalent thing in federal hiring or in USGS specifically?
Yeah, that's a great question. So my job has the title research as part of it. So that means we have a parallel process to tenure. So as a postdoc, I wasn't permanent. And then I actually got hired as a permanent person, but then I was just stuck at that level. Then I was able to apply and be converted to a research position. So for example, Lynn has the same type of position I do, where we're research people. So at the low levels, it's every 4 years you have to go up for review, and it's like a 10-year promotion packet. So it's like, but rather than being judged solely on papers, a lot of it's your impact. So you list references. So it's like, I can say Fish and Wildlife uses my research. I listed Grace from your program and somebody from Illinois DNR saying, these are state people who use my research. So then that's a committee that meets for about a week, and each person's assigned a couple of people, but then it's like they call all of your references and figure out what's your impact. Then there's a rubric that they go through to see what level are you doing research at? Like, what's the impact? Is it small? Is it national? So then that goes all the way up with different GS pay scales from an 11 to a 15. And then some of our superstars get promoted to a higher level. That's like the equivalent of a military general, or like probably like a distinguished professor or an extra distinguished professor. And it's also like Doug Johnson, who is affiliated, retired from that type of position. Let's see, Jim Grace, you may have heard of Don DeAngelis. I'm just rattling off the people I know. There's another USGS co-op person, Ronaldo Patino at Texas Tech. I'm sure Lynn would know other ST people as well. So yeah, there is a parallel process though. And people do jump back and forth between USGS and academia some. So because we maintain research programs, people are competitive.
Sweet, yeah, that's very helpful. Thank you. Sorry, go ahead.
I was trying to view the chat because I saw a really good question about women or comment. Let's see. I apologize. I'm bumbling around because we use Teams all the time. And so there was a comment that popped up about women not underrating themselves. And yeah, that's really important. So a couple of things about that. First, my center and more broadly, USGS has an equity, diversity and inclusion group. So we have a local group and also a national group where they really promote those ideas. Also, USGS, especially at our younger ratings, are about even split between male and female for research people. So older people, it's just longevity. They weren't, when they were hired 20 years ago, there was more, not as much balance, but it is a good spot to work because of that. Also, I went to a Fish and Wildlife Service meeting once, and I was talking to my postdoc mentor, and who was a guy, and then one of our other team members, she's a female scientist with USGS and experience. And we were just noticing that we were at a Fish and Wildlife meeting. It was actually mostly females who were there. And we were just wondering why. It's like, why are all these women working for Fish and Wildlife Service as scientists who have graduate degrees? And we realized it's because they have good work-life balance. And also, so like if you want to have a family and things, you're not hit as hard. So like Fish and Wildlife and USGS as well, we see a lot probably better balance than some parts of academia. I'm not sure, but work-life balance is another nice thing compared to academia as well. So, and also we get to have a paternity leave and maternity leave. So now it's 12 weeks guaranteed paid paternity leave once you've been with us for a year. So there are some strides that way as well. Also talk to Lynn about working as a Fed because she's a Fed and can give you perspective and your other co-op people.
Great. So we have time for one more quick question here. and I can read it off again. So how did you navigate the different hiring pathways in your career as a contractor, term, etc.? Any challenges or insights you can provide to early career scientists?
Let's see. Yeah, just apply. So it's like, get somebody to read through your resume. So as a contractor, I just sent my resume to Wayne and he interviewed me and selected me. So with the federal system, just what I talked about, like know how to apply. If you have like a grad student look over your resume, or if you're a grad student, have your advisor, or I hate to put Lynn on the spot, but have somebody like Lynn who's done it before, look it over. And this could just be like, yeah, so that's my suggestions there. And also Minnesota probably has a career service office, right? Todd or Chris or Nick?
Yeah, we definitely have CFAN's career services counseling that is very awesome. Highly recommend.
So you use them. And Chris, if you're in charge of seminar speakers, you need more seminar speakers, I bet they would do a seminar for you.
It's a good thought. I like it. Dr. Waterhouse has a nice comment in here about a new position as assistant unit leader of fisheries. And so check that out to anyone still in the Zoom call. I want to thank you again, Dr. Erickson, for taking the time to come talk to us about your career and also your research at USGS. And we will have another seminar speaker next week. So same time, feel free to tune in, everybody. And yeah, I will see you guys next week. Happy Friday.
Thanks, and I'll stick on for Peter.